So welcome back. We'll be continuing with the life cycle of Plasmodium. Okay, the life cycle of Plasmodium vivax, as we have already seen, it is digenetic. That is, it is completed in two hosts. One is in the humans, where a sexual phase is completed, while the sexual phase of the life cycle is completed in the female Anopheles mosquito. Okay, and humans, it is the intermediate host and the primary host is mosquitoes. Fine. The asexual phase completed within the humans, it is referred as a schizogony, which is a type of asexual reproduction that involves multiple fission. And during this, the nuclear divisions are completed before the cytoplasmic divisions begin. Okay, so it is not simultaneous. It is one is completed and then only the cytoplasmic division starts. The sexual cycle, as we have already seen, it is completed in the vector, or it is the primary host or the final host, and the female Anopheles mosquito. And here the sexual phase, it involves gametogony and sporogony. Gametogony, it is concerned with the production of gametes, the fertilization or the fusion of gametes, whereas the sporogony, on the other hand, it is post-zygotic multiplication, which results in the formation of the infective individuals, or the stage of the infective stage of the Plasmodium, which is referred as a sporozoid. I hope that is clear. Okay, now we will see in detail what is happening in the asexual phase of Plasmodium, life cycle of the Plasmodium. Okay, a healthy person acquires infection when a female Anopheles mosquito, which contains a sporozoid. Sporozoid is the infective stage. Okay, it starts from here. So the stage is known as a sporozoid, right? So here it is written. You can see sporozoids in salivary glands of the female Anopheles mosquito. Okay, so when the mosquito feeds on human blood, the sporozoid it is introduced into the bloodstream. Okay, so it is an infective stage. So it in the case of Anopheles mosquito, this infective stage resides in the salivary glands, and when these mosquitoes bite the humans for sucking up the blood, it introduces the sporozoid into the blood. So the mosquito punctures the horse skin by its proboscis of the salivary, what you call mouth parts, and first introduces some saliva into the bloodstream, along with which the sporozoids present in the saliva is also introduced or inoculated. And these sporozoids, they represent the infective forms of the parasite. And these are, we have already seen the sporozoids, the figure. This is the morphological feature. You can see it is spindle shaped, it is elongated, it is uninucleate, and almost measures up to like 12 micron in length and 0.5 to 1 micron in width. Okay, now what happens in the first stage? The first stage is the liver schizogony, or the part of the schizogony which is completed in the liver cells. Okay, that is the first stage of the schizogony or the asexual phase. Okay, now once within the human blood, the sporozoid circulates for almost like an hour, and then it gets into the liver cell. Okay, it gets inside the liver and that invade the liver cell. Okay, it gets inside the hepatic cells, that is a liver cell, right? And now starts the what you call asexual multiplication by way of schizogony. Schizogony in liver cells. After penetrating the liver cells or the hepatic cells, each sporozoid grows for a number of days and then it becomes spherical in nature. You can see this is the spherical transformation of the what you call the sporozoid. Okay, and this spherical phase, it is non-pigmented and is referred as a schizont. So sporozoid, it passes through blood for a couple of hours and then it reaches the liver. It enters the liver cell, and once it is inside the liver cell, it gets transformed into a, this is this is the liver cell. Okay, and this is this what you call the plasmodium, right? So it becomes spherical in nature, non-pigmented, and this stage is known as a schizont. Okay, now this schizont, it divides by schizogony or multiple division or multiple fission, right? And produces a large number of uninucleate cells which are known as cryptozoids. You can see like there are plenty of uninucleate cells which is formed by the multiple fission of the schizont, and these cells which are produced by the multiple fission of schizont, it is known as a cryptozoid. Okay, or it is otherwise known as cryptomerozoites. Okay, cryptozoite is enough because that is what is labeled over here. Okay, cryptozoites. Now the cryptozoites are liberated from the liver cell uh, when the liver cells burst. You can see here the liver cell, it bursts open and it releases the cryptozoites. These cryptozoites normally invade like new fresh liver cells and undergo schizogony, just like what happened over here. Okay, so the cryptozoites had entered the liver cell, a new cell which is not infected till now, and then it develop into schizone. Schizone undergo multiple fission, it produces cryptozoite. So this is like it is what you call continuous. Okay, but what happens is these cryptozoites which are produced from the cryptozoites, they are referred as metacryptozoite. Okay, I'll repeat it. Here cryptozoites which are produced invade fresh liver cells, undergo schizogony similar to the previous one over here, and produces a large number of metacryptozoites. Okay, so here it was cryptozoites, and this one is known as a metacryptozoites. They are also referred as phanerozoites. Okay, metacryptozoites. This may be repeated several times. That is, these metacryptozoites may again invade a new fresh liver cell and continue the phase like this. Okay, the new liver cells are infected each time. Okay, all these succeeding schizogonic divisions are referred as exoerythrocytic phase. Okay, exoerythrocytic phase. So the whole phase is known as exoerythrocytic phase. The exoerythrocytic phase of the plasmodium, it remains immune to the resistance of the host. So that means the immune cells cannot actually uh, uh, break down these cryptozoites, or it cannot actually bring down the number. Okay, and the parasites and are not susceptible to the action of any kind of anti-malarial drug as well. So what happens is at this particular phase, the plasmodium is actually not susceptible to any kind of resistance. Okay, now the next phase starts. That is, the here what happens is these cryptozoites, it can then enter into the erythrocytic schizogony. So the part of the schizogony which is completed within the RBC is what is referred as a erythrocytic schizogony. These metacryptozoites, after escaping into the blood, that is after the bursting out from the liver cells, it may get into the blood, and these metacryptozoites, it may escape into the blood and then it invade the RBCs. Okay, each of the cryptozoid, it becomes rounded and modified into a young trophozoite. You can see here, while it is moving through these metacryptozoites, unlike the cryptozoites, they are slightly elongated, and these enter the RBC. Once it is inside the RBC, it has taken again a spherical shape, and this stage, which is inside the RBC, spherical in nature, it is known as a trophozoite. Okay, trophozoite, we have already seen the structure. Okay, trophozoite inside the RBC. So this is the phase which we can actually uh, look into for the presence of plasmodium in the body. Okay, so when we collect the blood and if we study the what you call blood smear, this is how uh, the trophozoite infested RBC will look like. Okay, so this is what we are. Okay, so the trophozoite. Now, this trophozoite, it grows in size, and as it grows, a central vacuole is developed. You can see here. So this is the phase passing by. So the trophozoite is there, and it grows in size, and as it grows, a central vacuole is formed. Okay, central vacuole is formed, and cytoplasm is pushed towards the periphery. So it seems like a ring. Okay, and the nucleus is pushed to one side, the cytoplasm is pushed towards the periphery, and this stage, it resembles a ring-like structure, and this stage is known as a signet ring stage. So signet ring stage is developed from the trophozoite stage of the plasmodium, and it takes place inside the RBC of humans as a part of the schizogony, erythrocytic schizogony. Okay, it resembles a signet ring with a peripherally located nucleus looking like the You can see here, this is the what you call the nucleus. Okay, so this resembles that of a gem of the ring, right? And this stage is actually not present in the Plasmodium falciparum, but it is a significant stage in the Plasmodium vivax. Okay, now this signet ring trophozoite, this one, it will ingest a large amount of cytoplasm of the red blood cells. Just 1 minute. Okay, it actually takes up cytoplasm of the red blood corpuscles, forming a food vacuole into which it secretes a digestive enzyme. And the enzymes bring about the proteolysis. Okay, the enzymes which are present inside this one, it will bring down, or it will break down the hemoglobin, and it will produce break down the protein component of the hemoglobin, and hematin is produced, and this protein is used as food by the trophozoite. Okay, so the cytoplasmic contents are being broken down with the help of the digestive enzymes, and these enzymes also break down the blood hemoglobin by way of proteolysis, which break down into the hemoglobin is broken down into its protein component and hematin. This protein component, which are produced by the breakdown of hemoglobin, it is used as a food by the trophozoite, while the hematin, it forms a toxic material, toxic pigment, which is known as the hemozoin, which gets deposited in the food vacuole. Okay, you remember this one, isn't it? It like remains inside as hemozoin. Now the signet, now what happens is the signet ring stage, stage of the trophozoite, it will develop into an active amoeboid trophozoite. Okay, you can see here pseudopodia like structures are formed, isn't it? So this is the amoeboid trophozoite, and at this time a small red eosinophilic granules appear in the cytoplasm of the host corpuscle, that is the host RBC. It develops a certain eosinophilic granules inside the cytoplasm, and these are known as, you can see here, Schaffner's granules. So Schaffner's granules are actually the red eosinophilic granules which develop inside the cytoplasm of the host RBC. Okay, and these are developed at the what you call the amoeboid trophozoite stage in the schizogony. Okay, these amoeboid trophozoite, they continue active feeding, and it becomes rounded. You can see over here, it becomes rounded. It grows in size and becomes a schizone once again. That is, a schizone means a stage which is ready to undergo a schizogony, multiple fission. Okay, so it becomes this amoeboid trophozoite, it undergoes transformation into a spherical schizone stage. Okay, now this schizone, it undergoes schizogony. What is schizogony? Multiple fission. Okay, its nucleus divides to form 12 to 24 nuclei. You can see here multiple nuclei here. Okay, and each of these get arranged at the periphery, and cytoplasmic mass surrounds them. So you can see here RBC may contain smaller uninucleate cells like this. Okay, so each of the cytoplasmic mass with one nucleus, okay, it is called as a merozoite. You can see here. So here actually how many are there? 6, 7, 8 merozoites are there. There can be plenty, like 12 to 24 merozoites may be present inside a single RBC. Okay. Now the hemozoin granules, here you can see the hemozoin granules, they get gathered at the center. Okay. This is followed by the rupture of RBC and the release of merozoites into the blood plasma. So the RBCs gets ruptured, and all these merozoites which were confined within the RBC, it gets liberated into the blood plasma. These released merozoite, now it can invade new fresh RBCs, that is which is not infested so far. Okay, a fresh RBCs may be invaded by these merozoites to repeat the erythrocytic schizogony, right? At the same time, these ruptured RBCs, which are referred as ghost cells, these ghost cells, it may get destroyed, it may be taken into the spleen. Okay, and one complete erythrocytic schizogony, it takes place, erythrocytic schizogony, it requires 48 hours to complete in the case of Plasmodium vivax. Okay, so we had pre-erythrocytic schizogony or exoerythrocytic schizogony, we had erythrocytic schizogony, and then we have the post-erythrocytic schizogony. Sometimes what happens is these merozoites which are produced, reach the liver cells. You can see over here, it may reach the liver cells and then undergo the schizogony development in liver cells. This is what is referred as a post-erythrocytic schizogony. On the